Good day, everyone. My name is Ken Buetow. I'm the Director of Computational Science and Informatics from the Complex Adaptive Systems Group at Arizona State University. I am a professor of School of Life Sciences as well. And in today's talk, I'm going to be presenting information on how we can embrace the complexity of cancer and how through this embracing of the system's approach to cancer, we can get novel insights not previously obtainable by traditional approaches to examining the problem. As this group is more than aware, we're in the middle of a tremendous transformation in biomedicine. That transformation is embracing the concept of personalized medicine. Personalized medicine being delivering to the right individual, the right care at the right time and in the right place. This transformation is both revolution, as we now have the capacity to look at the very molecular nature of an individual and their disease, in particular in cancer, in tumors, but this personalized medicine transformation is also a renaissance, where we have the capacity to not only look at the molecular characteristics of a person, but all of the rest of the clinical and non-molecular components and integrate those in a manner that facilitates us getting a novel perspective on the disease and a whole that's greater than the sum of the individual parts. This personalized medicine revolution actually isn't completely new. And in fact, as with many things, the future is already here in many places, not yet evenly distributed. For example, this from an article and an ad in a recent Washington Post, but in fact you can find this in almost any type of lay presentation nowadays, is an offering of the ability to do molecular characterizations, in this instance from our colleagues at Vanderbilt University, to help direct treatments. More specifically, they've created a resource called My Cancer Genome, shown here on this slide. Materials shown here courtesy of our colleagues at Vanderbilt University, Maya Levy, where basically one with a particular cancer that's been molecularly characterized. You can see here one can select your particular cancer type. In this instance, we'll choose melanoma. Once we choose melanoma, we can see a particular type of molecular characterization for a given gene, here showing the BRAF gene. If we see within our BRAF gene we have a particular form of variation, this type of infrastructure now lets us decide what type of treatment to get. So I realize, well, this is difficult to view. Let me blow up at the bottom. So with an individual with this particular flavor of V600E mutation, one can actually see what the function of the mutation is and go through this and select particular types of agents that have been characterized to perhaps have differential response in individuals with this type of disease, this particular molecular form of cancer. And then from this, then, figure out what types of interventions to take. So this is very exciting and a novel approach to looking at cancer. But I guess I want to indicate that at best, this is the tip of the iceberg. And more pragmatically, cancer itself is probably, and well established at this point, to be way more complicated than just a collection of point mutations, and that truly understand cancer, we need to probably look at the disease in its entire molecular context. So what do I mean by this? Well, cancer is a complex adaptive system. And I'm going to spend a little bit of time in the next vignette walking through what I mean by using complex adaptive system in a molecular sense, and then I'll use it in a formal sense. First, we know that cancer occurs at a somatic level, at the basic tumor level, by the acquisition of multiple mutations that ultimately transform normal cells in a compartment of a normal tissue into a tumor. It's now well known that these mutations, actually there are hundreds of these mutations occurring, and that these mutations don't occur at random. They actually occur in a particular context and actually transform the particular collection of cells in this organ into a new organ, that there's three-dimensional structure to this, and that these collections of mutations essentially mimic the sort of the standard evolutionary process that would occur in standard speciation. Basically, what one has is the de novo generation of a new organ within an organ. Unfortunately, this new organ isn't constrained by the traditional multicellular contract and has devastating impact on one's health. But over and above this, what we know is that these alterations, these hundreds of alterations, can occur not just by point mutations, for instance, in individual genes or in individual cells, but in fact there's a rich collection. Cancer has a very large tool chest of alternative ways that can actually generate these hundreds of alterations. So it can do it via mutations. It can do it by changing the copy number, amplifying big chunks of DNA, so that in fact you get the moral equivalent of having mutated a gene by either removing it completely and its neighbors, or by rapidly or dramatically expanding its content so that it has the appearance of being turned on, even when in fact it might not have had an formally activating mutation. We know that you can actually have alterations that change the expression of genes, that can actually be, the genes can still be of normal structure and function, but their expression has been changed. Again, either radically increased or radically changed at the RNA level. We know that there are non-nucleic acid-based means to actually create these alterations, epigenetic modifications that change not the specific nucleic acid structure or the quantity of the nucleic acid structure, but how the portfolio of changes. These are through changes of chromatin configuration, through a variety of complex processes. Not only through epigenetic processes, but we can actually change the particular portfolio by microRNAs. Now a great deal of enthusiasm and interest in what was previously called the junk DNA, the other 98% of the genome, appears to actually not be inert, but in fact has the capacity to have dramatic changes on the outcome of a particular system. As well as there can be non-protein, non-nucleic acid means that actually change or can generate this portfolio of alterations. So what one sees is it's not just simply point mutations accumulating in individual cells. There's a complex collection of molecular processes that can also drive the outcome. We also see that, in fact, these hundreds of mutations are not occurring at random. These hundreds of mutations are targeting, and again, several decades worth of biology, this described probably more than 15 years ago by Hanahan and Weinberg, indicate that there's a number of biologic processes that must be evaded or altered if we're going to develop cancer. Growth factor independence. Tumors need to evade apoptosis or programmed cell death. They need to maintain their telomeres so that they don't go into senescence. They need to work to avoid cell cycle regulation, and they need to continue to feed themselves through angiogenesis or other processes. So we can see from the get-go that it's more than just a single alteration that was such as we saw in the earlier portion of this that are critical. You need multiple alterations in multiple processes in order to be able to develop a tumor. Well, what we also know is even as simple as this sounds, these individual processes are actually complex networks of interacting components. So if we look at these, we can see there are multiple, sometimes dozens to networks that may, that may have as many as 100 different interacting components that can all be altered in one manner or another to generate the specific phenotype of cancer. And what we see is that within these processes that a given individual may have an alteration in one set of genes, whereas another individual may have an alteration in a different set of the genes, still targeting the same process, but no longer the same collection of mutations. Or yet another person can have another set of alterations that can generate this process. So we can see that the underlying molecular mechanics of the generation of tumors, while it may require hundreds of individual components, those components are non-randomly distributed through a complex collection of networks. Lastly, what we know is that these germline mutations that occur, or these somatic mutations that are occurring in an individual tissue also occur in a context. And that in fact there is normal underlying variation in one's genetic constitution that can also alter these processes. So in addition to the hundreds of somatic mutations, there are millions to tens of millions of variants that are part of our normal genetic constitution that vary from the DNA I received from my mom versus the DNA I received from my dad. And these variations in normal genetic constitution can drive how one performs angiogenesis, the structure of one's cellular matrix, the specifics of one's immune response to both endogenous and exogenous exposures. These variants can also have a dramatic effect on how we react and respond to our environment. They can decrease, they can amplify, they can eliminate, they can change the portfolio of response and how a host sees chemicals, viruses, their own internal hormones, or one's nutrition. So what we can see is that while we are at the beginning of personalized medicine, particularly at the molecular level, that the presumption that a single mutation in a given gene is sufficient to drive the full oncogenic process, and elimination or mitigation of that may be a bit of an underestimate. And that in fact cancer is not only complicated, it's very complicated. A number of complex molecular interacting components. So cancer is a complex adaptive system. So what does that mean? Well, let me walk through a more formal definition of this. If you retain any key concept of what is a complex adaptive system, the key piece to remember is the whole is more than the sum of the parts. That simply reducing the system to its individual parts will not capture its complexity. And that there are what we call emergent properties, that there are features that are properties of the system that are not simply described by only examining its individual parts. And this is very important as we embrace future study of cancer. It means that simply only applying the reductionist approach of looking at individual components may not lead us to being able to truly understand cancer or to do its interventions. So let me drill down a little bit more. So that's the key essence. So a complex adaptive system evolves over time. We could see from the molecular diagnostics that we certainly meet that criteria in cancer. It has adaptive behaviors. So, and as we're all aware from those of us who are familiar with or have ever been exposed to individuals treated with cancer, that cancer is a dynamic entity that changes in response to how it is touched. It displays emergent property. And that, in a certain sense, not only does this complexity suggest we may not be able to understand cancer simply by assembling the individual parts, but it also means that because it has emergent properties, there is the potential for unintended consequences, that it's not just simply A plus B equals C, that intervention can have changes that we actually won't understand until we look at it as a system. Consequently, to all of the above, it requires transdisciplinary study to understand its multidimensional interactions that include all the fields of biology, all the fields of biomedicine, and a rich ecosystem of interacting parts. I think the key thing, though, that I want to focus out of this is that cancer, because of its complex nature, has interdependencies. That looking at any individual component in isolation won't tell us the whole story. And that these interdependencies are key in two dimensions. They're key in the dimension of the resources that they require, and they're key in the context of information. And what I'm going to focus on in the next section of the talk is how do we go about studying and evaluating these interdependencies? How do we go about understanding these complex relationships? So shown here is a graphic that shows a standard network. And in fact, it turns out that most of natural phenomena occur and are engaged in network class phenomena, whether it be something as artificially created as the internet, as to the biologic networks that I just described earlier in terms of driving particular phenotypes. Our somatic genetics colleagues have long now been forced into understanding and looking at cancer in the context of network models. And it's basically because when one did the molecular characterizations that I described at the beginning of the talk, the hundreds of mutations were observed. It was observed that, in fact, while any given cancer may have hundreds of mutations, if we look across cancers, most of those mutations are not recurrent. They have a very low frequency of recurrence, most of them less than 5%. Even the most common recurrent mutations rarely occur at greater than 5%. We also observed that there were variation in gene signatures. If we looked at the transcriptome, what we saw is that, that while there are the ability to see common patterns, that almost every individual has a unique transcriptome. I think most disheartening, though, is that the multidimensional data that was emerging from large-scale projects like the Cancer Genome Atlas that showed almost conflicting, canceling results. One couldn't find coherence. One couldn't find a consistent pattern of outcome that emerged as a product of looking at these multidimensional cancer molecular characterizations, similar to what I showed at the beginning of the talk. So what the somatic genetic community has emerged is the recognition that it takes a network. And actually, as we look at these networks, we can see that patterns of coherence start to emerge. So even though, for instance, this is from the early work of the Cancer Genome Atlas project, even though we were seeing hundreds of mutations, these mutations actually collapsed and had recurrent patterns if we assembled them against networks. Shown here are three different networks that emerged as being important in the development of glioblastoma multiforme, the very common form and unfortunately still quite lethal form of brain cancer. We could see that if you looked at alterations within these networks, that, for instance, the RAS signaling pathway, the RAS PI3 kinase pathway, was altered in 83% of GBMs. When we looked at the P53 network, or looked at multiple members of the P53 network, 87% of those were 80% of individuals with glioblastoma had an alteration in that network, and slightly lower frequency, but still very high group of associations is if we look at the retinoblastoma network, signaling network. Again, 78% of individuals had alterations in that network. So my group has been interested in coming up with formal ways. These have just been sort of descriptive ways of assembling this information. We've been interested in approaching this in a quantitative manner. And so shown in the next couple of slides is some specific work done by my group to identify key processes underlying cancer by focusing on networks, but in a manner that allows us to objectively see the alterations that are occurring, as opposed to descriptively finding those. The first piece of work I want to describe is generated by a postdoc that worked for me, Saul Afroni, who's now a faculty member at Bar-Ilan University in Israel. He developed a new means of examining, computationally examining cancer by looking at the molecular state of particular networks and or their interactions, and developed a tool that allows us to use that state information to look for non-random patterns of alterations. This pathologist tool has been now, the paradigm of this has been captured in a new tool developed by Vasquez et al. That's actually in widespread use now throughout the molecular community, the TCGA community, called Paradigm. Basically, the idea of this type of analysis is that we actually look at the network as a computable entity, something that we can say is processing information, just the same as a computer or a computer program processes information. Shown here is a diagram of a network, an artificially constructed network, that has different components. You can see on the edges or the outsides of the diagram things called A, B, C, F, and G. These would be genes, and we can measure their particular expression levels, as shown at the bottom of the slide, measuring whether they're present or absent, or a quantitative measure of their level. Then what you see is the black entities on this graphic. The black entities with circles are places where the genes interact to produce or change the state of either themselves or some entity downstream. So at these interactions, we can assess whether a particular node or a particular product is either active or consistent. And let me walk through how we do this. So we take a network diagram such as this, we overlay the specific pattern or the state of genes as shown in the next slide. So here we took those individual genes, overlaid their expression pattern, and blue means that the gene is present or on, red means that it's absent or off or up or down. We can use a variety of different ways. And what we can then do is walk through each of the interactions. Let's start with interaction 1 and look at genes A, B, and C. What we can see from the symbols of this particular interaction is that if A is present and B is absent, we should get the product C. The red blockage on B would say that if B was present, it would inhibit this interaction. If A is present, it should activate or produce C. So what we can do is look at this and score and say, is this active? Well, yes, and it's consistent because it produces C in the absence of B. Let's try this again now with interaction 2. We go to interaction 2, and we can see in order to produce D, or to have interaction 2 be active, we need to have both A and B be present, or they both would need to be blue. So if we score this, we can see then, no, 2 is not active, or 2 is not consistent. We shouldn't be seeing D because we need technically A and B to be present in order to have the product D. One last computational step. So let's look at 3, where we need to have E and D present, where we need to have E present and D absent in order to see F. And in this instance, we can see that D is present, so we should not see F. D should inhibit the presence of F, and in fact, that is what we see. So we can score this as inactive or consistent. So by using these network structures, we actually change the dimension of our quantitative or computational analysis of the network and of the data. So we're no longer looking at individual gene states, but integrating the information as described by this biologic program and looking for differences in patterns. So what's the impact of this? Well, first, it helps us deal with the huge dimensionality problem that we face in doing biologic analysis. So it reduces the dimensionality and complexity. So rather than looking at a standard gene signature where we could see for this particular data there would be 7 signals that we needed to be managing, we can see is by scoring pathway consistency, we could go as low as 3 if we look at just the interaction, so we have a more than 50% reduction of complexity. Or if we actually assemble this and treat the pathway as a unit, we can see that we go all the way from 7 to 1. So we've reduced the complexity now by 7-fold, which gives us more statistical power to look at the biologic phenomena. And if we go back and remember that much more complicated diagram, we can now look at processes like apoptosis, or look at processes like growth factor independence, and treat them as single entities rather than having to inherit the noise associated with the individual genes. What we also see, though, is that while these may reduce the complexity, they can also reduce some of the noise or the differences. This is now overlaying a pattern on this particular network that we could see if we're scoring consistency again, that if we looked at this in the raw gene signature space, we'd see that there are 4 differences between these 2 individuals, whereas if we look at pathway state, especially even at the interactions, we go from 4 to 0, or conceivably, no differences at all in this particular data. Interestingly, though, by looking at pathway consistency, there are instances where we amplify the differences. So we can actually measure completely the effect of this particular network. So in gene signature space, scoring just the individual pieces of the genes, we can see that we have only one difference between the 2 individuals that we're examining here, whereas if we look at the pathway consistency, we can see that there's actually 3 differences. So this gives us a very different approach to examining the problem and seeing different flavors of outcome and different flavors of answers. We've also been interested in applying this not just to somatic data, so that, as I showed in the previous work by Saul, but also in constitutional variation, that millions of variants that we inherit from our mothers and fathers that can have profound differences and profound impact on our individual phenotypes. This work was done by a postdoc of mine, Rosemary Braun, who's now at Northwestern University, and leverages a technique that she developed called pathways of distinction analysis, a way to look at constitutional variation and understand how those patterns of constitutional variation may differ between individuals with alternative phenotypes. What I'd like to do is focus on some of the outcomes of the work, a specific example that shows why approaching the problem from this network perspective may be important. And it leverages work that was published by Rosemary that leveraged a published data study by Hunter et al. That was published in Nature Genetics in 2007, looking at a large-scale genome-wide association study, where in the primary work they looked at over 550,000 SNPs using an Illumina array and found a handful, 1 or 2 genes that, or 1 or 2 SNPs that turned out to be significantly associated. What Rosemary did in this analysis is, instead of looking at the SNPs individually, she looked at almost 1,100 pathways and looked to see how the pathways differed between individuals with cancer and individuals without breast cancer in this instance. Just to point you to the key and interesting findings shown here is a network diagram. We can see how names of individual pathways may be as misleading as names of genes. So if we look up at the top here, we can see the name of this pathway is prostate cancer. In fact, it's a hormone regulation pathway related to FGF. So that in particular is not necessarily the names of these pathways, but a key finding of the original Hunter et al. Paper was this gene here, FGFR2, was significantly different between individuals with breast cancer and individuals without breast cancer. It's the most significant association. It's been repeated in a number of individual studies. What was observed, though, is if you look at this, we can see that FGFR2 is actually embedded in a very complicated network. And in Rosemary's analysis, we not only looked at, in this instance, FGFR2, shown here with the red circle, but we also looked at all of the variation in the other genes in the pathway. And what became interesting to us is we saw that, of course, here's the finding from the original results, FGFR2, highly associated, although with a small relative odds ratio. We found that, of course, we could find that that pathway was important when we looked at the pathway, including the FGFR2 variation. But what was interesting to us is not only we saw that when we looked at the pathway when we looked at the pathway in toto, we found a slightly larger risk, still very highly significant, odds ratio of 1.38 with a p-value of p to the minus 10. But what was interesting is when we looked at this, if we dropped FGFR2, the very finding that Hunter et al. Had, we still had a significant association. In fact, an odds ratio of 1.32 with almost the same significance. So what we can see is it's not just FGFR2 that's driving the difference of whether one develops breast cancer or not, but also variation, other variation in other genes in that network that are also contributing to these patterns of variation that may be causing breast cancer or may be important in determining who gets breast cancer. The significance of this, of course, is that it gives us alternative targets and may also suggest to us that it's not just a single mechanistic approach, but that in fact nature and or cancer can leverage multiple different variants in order to achieve the same goal. What I wanted to show in this is where does the overall significance of this particular pathway say? So this is we're back to our shown with the arrow here is what the FGFR2 retaining pathway. We can see, interestingly, it is a significant pathway in Rosemary's analysis, but over and above that, it's by far not the most significant pathway. We can see that purine metabolism, calcium signaling, and other pathways are also important in producing breast cancer. And in fact, there is other epidemiologic literature that, of course, would suggest that purine metabolism and and calcium signaling should and could also be important in developing cancer. And in fact, we find statistically strong evidence that that is indeed the case, and find evidence for more than just the small number of variants that were identified in the original study. The other thing we can do is actually create, not looking at the networks just as individual, but actually now start to assemble the effects of networks. And when we do this, what we can see is if we look at the original odds ratio, so again, the Hunter et al. Odds ratio was around 1.3, we can see if we actually look at the top two pathways, we now actually start to get much more significant odds ratio. So if we look at the top two pathways from Rosemary's analysis, and note this does not even include the FGFR2 result, we can see that we have an odds ratio instead of 1.3, we have an odds ratio of 2.02, so twofold excess risk rather than about a 50% excess risk. And we can see if we assemble across all of these networks, we get almost a 3.5 to 4-fold excess risk of developing breast cancer by looking at the entire network of susceptibility factors, not just a single SNP. And it's useful to put this into contrast of the recently published Nature article, where even with looking at huge numbers of studies and combining all of the individually significant SNPs, the maximum odds ratio that they reach for the highest 5% risk is 2.4, and what we can see is even with all of these large scale and segregating to just the smallest number of high-risk loci, we only then begin to approach the findings that Rosemary found by looking at the individual network components. So we can see that the data that we're approaching in cancer is complex. We can see that by doing large-scale genomics, the data is large. And I guess part of what I want to do is start to transition into the talk from talking about how we can mechanistically embrace the complexity of cancer and how we would generate and utilize this information using novel algorithmic approaches, is also talk about embracing the other complex dimensions. So this is embracing the revolutionary piece. How might we also embrace the other components of cancer to embrace the renaissance piece, the ability to integrate and utilize all of the information in the system, just the same as our analytic approaches, or leveraging additional information in our approach to the disease? Well, first thing we need to know and embrace is that personalized medicine is a big data problem. So what do I mean by big data? I know there's tons of description nowadays. You actually can't open a newspaper or magazine or hear a TV show. Even the most recent presidential elections in the United States captured or leveraged big data approaches to problems. So let me spend a minute defining what is big data. Well, big data is both, not surprising, big. It's large in volume, and certainly in the context of biomedicine, large-scale sequence data is very big. But big data, as defined in the emerging new field, has a large variety, which you can see we're seeing and is problematic in our cancer problems, but also changes rapidly and needs to have the capacity to do dynamic computation. So in approaching big data, what we realize is that it's a multidimensional problem itself, not unlike any complex adaptive system. Well, unequivocally, the genome itself is big, and we can see computationally intensive. So a given genome, a sample for a given individual, the raw data, the FASTQ data that comes out of a sequencer, is over 200 gigabytes of raw material. And even when processed, collapses only down to 100 gigabytes. So we get a twofold reduction. And even if we reduce, to be honest with you, to just the essence of what differs without throwing away a lot of the primary observational space, we're still at 100 megabytes per sample. It's also computationally intensive. We can see that in order to do the processing that I described above, we need to have hours, sometimes days, of computer time and dozens of computer processors in order to achieve our results. So again, it's very large in just processing the raw molecular data. However, the genome and the molecular data is but one dimension in our multidimensional problem. The genome, as said before, is complemented by the phenome. We know a lot about clinical information, as well as the exposome. And each of these individual sources of data is at least as large as the genome. Well, what do I mean by this? Well, the phenome data is spread out and may be a longitudinal health record that captures all the information across an entire lifespan for an individual, and is quite large and is distributed, meets this very large and distributed and complex and heterogeneous definition of big data. But it's also voluminous in other dimensions, the phenome data. So again, as large or larger than any of the genome data resources, we can see our biomedical imaging and increasingly the capacity to do non-invasive characterization of disease and disease states or specific interventions. This is actually looking at what happens with radio-labeled tagged chemotherapeutic agent over time in an individual. Each one of these individual scans is as large or larger than a human genome sequence. And of course, this is but the tip of an iceberg. We are seeing unequivocally that the explosion of the monitored self that's available through real-time mobile devices or personal devices that are increasingly ubiquitous throughout the health ecosystem are going to dwarf and require us to have the capacity to process incredibly large volumes of data. So how should we as biomedicine individuals and investigators in biomedicine embrace this problem? What can we do? What should we do? Well, I would argue, following the advice to Benjamin and in the graduate student or in the graduate, I think actually the future for us in biomedicine can be simply borrowed from a page of a very highly successful current art activity. I would say I have one word for you all: Google. So Google deals with big data, large-scale pattern identification and analytics in real time, all day, every day. And what we need to be doing in biomedicine is approaching the problems like Google. Now, what do I mean by this? Most people, when they hear Google, and actually quite appropriately so, think in terms of search and the search engine. What I'm talking about here, though, is not actually the search capabilities per se of Google, but its big data capabilities to actually process and interpret data. Its capacity to find patterns and utilize those patterns in real time. So shown here, and actually all of us are familiar nowadays in using the Google search engine, is as soon as we start to type, Google starts relying on the complex underlying patterns of data of literally tens to hundreds of millions of individuals to understand what we may be trying to do and look for non-random activities that it uses then to facilitate and helps it understand what we're attempting to do. We need to be embracing similar such approaches and analytic capabilities in our universe in biomedicine. Another famous example that probably all of us have encountered is Amazon.com or Amazon. So I guess if I had a second word for you, it would not be Google, it would be Amazon. So Amazon. Well, we're all familiar that when we actually engage the Amazon website, in this instance, if I'm searching for a book, this instance too big to know about big data, Amazon analyzes the non-random pattern of other individuals that are part of the activity and that have presented and done similar sort of searches, and then uses that information to try to identify other books or other related activities that I might be interested in performing, and other types of findings that I might be interested in. And we can use this similar sort of approach in our approach in biomedicine and in analytics across the biomedical space. So this is all part of an emerging new fourth paradigm of science, data science, as it's being labeled. So fourth paradigm science or data science complements the traditional data-generating science with data-driven science. And in fact, commonly when people talk about fourth paradigm science or data science, the definition on the second bullet here, instead of saying data-generating science, would say hypothesis-driven science. I refuse to surrender the concept of hypothesis-driven science to data-generating science, and would argue that my colleagues in astronomy, physics, and economics actually are all still doing data science and can do hypothesis-based science without actually generating or doing de novo experiments. To the best of my knowledge, we have a pretty good confidence in planetary motion without ever anyone physically moving a planet to date. So I think that there is validity to data science, and with the appropriate scientific construct, the difference is whether we're generating data versus leveraging observational data that may already be in place. So what are the characteristics of data science? Well, it's data generated by instruments or simulation. We actually know from the previous talk, we're generating tons of data in the molecular space, processed by software, and the information and knowledge is stored in computer, and the scientist analyzes using data management, statistical, mathematical, or algorithmic approaches. So the experiment is actually conducted in the computer using large data reservoirs, and it's ubiquitous in other fields of science, and we would actually argue is critically important in biomedicine, and that biomedicine move into this space as well. The other attractive part, I guess, of being a data scientist is, as the Harvard Business Review indicates, it is an up-and-coming field. And I have to say, for probably the first time ever in my life, I would be considered to be doing sexy work by doing data science. I think maybe this falls into the universe of geek chic that's emerging. So it is an up-and-coming field, and so if you're looking for career opportunities, data scientist is a new place for you. My group's been very interested in approaching this large-scale biomedical space through the creation of a new data science instrument. And that science instrument we're calling a next-generation cyber capability, or NGCC. So What differentiates a data science instrument from a traditional computational approach is that it's, well, first off, we're very familiar with data generating scientific instruments. What we're interested in doing is generating a data science instrument that's composed of physical capacity, an elemental whole that combines ultra-high bandwidth networks with large-scale storage measured in petabytes, multiple flavors of computation, not just high-performance computing or transactional computing, but also embraces the new paradigms of big data computing. We're interested in an inherently and elementally engages logical capabilities, unique software, the management of data about data or metadata, and the management of information about data, the semantics, the interrelationship of how, so it's more than just data about data, it's how the data is interrelated to each other. Lastly, our instrument that we're trying to create at ASU is complemented by having, and again, elementally composed of transdisciplinary teams that know how to do high-performance computing, know how to use ultra-high-speed networks, that actually know how to generate next-generation algorithms, and are not intimidated by managing petabytes worth of data. Shown on this cartoon is just the assembly of that elemental whole. We can see that at the bottom of this slide, we have the computational capacity, high performance, massively parallel, embarrassingly parallel jobs that can perform traditional high performance computing capabilities. We also then have big data capabilities similar to the high performance capabilities that allow us to approach the problems in this instance from a big data perspective as opposed to traditional high performance computing. And then we have this analytic capabilities, this logical capabilities shown at the top in the big box here that allow us to manage both the context of data as well as analytic and data management resources that we can see here on this particular slide. Key to this activity, though, is the rich content. And what we're assembling in our large-scale data instrument is a diversity of types of biomedical information, from case report forms to instrument-generated data to electronic health records, the whole cornucopia of rich types of data that are and can be accessible. So our view in this is that the key characteristic of the NGCC, or data science instrument, is that it connects all of these different types of data and resources in a unique environment, and within that environment allows us to step out of the hell described by T.S. Eliot, where nothing is connected. It allows us to leverage these unique resources in novel ways. So we have a couple examples of how we're already using this, how the NGCC can serve as an evidence engine by assembling all these different types of data, and how we can actually use this in a manner not unlike Google might use the data or use such an infrastructure. So in this particular instance, we leverage work done while I was at the National Cancer Institute, leveraging raw data, in this instance of imaging data. Shown on this slide are a bunch of imaging characteristics, and what we can do is, leveraging the NGCC class infrastructure, is look at the individual characteristics of a single patient. Think of it as your personal preferences in ordering a book, or the words that you've typed in to date into your Google search as it's trying to figure out what flavor of search you want to do. Then, based on these characteristics, we can pull from this large, complex, heterogeneous data resource, we can see what would be the outcomes of patients that have those particular characteristics. So we can actually choose and select patients and use individual patients like me to see what might be the outcome of a different intervention, or just for you as an individual with these characteristics, how do you compare to the average survival of an individual. In this instance, we were looking at glioblastoma. How does your survival with your characteristics compare to the overall general glioblastoma survival? And we can see in this instance, not radically different, which we can argue is either positive or negative. The prognosis is not worse, but in this instance, the prognosis isn't formally better. We've also used this to explore big data technology proof of concept by assembling molecular sources of diverse sources of molecular data from the Cancer Genome Atlas, the NCI Nature Pathway Interaction Database, the NCI Cancer Gene Index, and other information on gene drug database information that we have available to us to assemble across these problems. So in this type of examination, what we can do is ask simple clinical research questions, like for tumor samples which are from patients who've had progressive disease. What are the genes that are high amplification areas? What are the genes that have actually shown a difference in their copy number alteration? We could ask queries such as retrieve the gene expression scores for all the genes that share a high amplification region and find out how that changed chemotherapy response. Does it alter chemotherapy response? Or lastly, we can look at genes that have a documented association with one or more drugs and see if they are in known amplification regions. So we've actually done these test queries in a proof of concept of this particular type of work. But what's exciting to us is not this particular clinical research questions, but actually the potential big data questions that would feed not just clinical research, but overall clinical outcome, like given clinical, demographic, and lifestyle characteristics of this patient, how does survival differ among alternative interventions? What are the adverse events associated with particular alternative treatments? Are there quality of life differences associated with these alternatives? And more pragmatically, and perhaps full circle in our conversation of Renaissance, what might be the right intervention for me, given both my molecular as well as other clinical characteristics? So, in summary, we're approaching in our group cancer as a complex adaptive system. And we think that it helps us address some of the challenges that the cancer community is currently facing in both understanding disease and hopefully directing new development of therapeutics and interventions. We believe it's technically feasible to create and deploy technology to exchange information within and between members of this complex ecosystem. And we believe that new network models will help us augment the sort of genome organization models to manipulating and understanding data. And lastly, that if we're going to approach cancer as a network, we're going to need a multi-stakeholder, multi-dimensional community to address these problems. So with that, I'm very interested and happy to take questions. Please feel free to enter your questions into the question block. I will actually go through those questions, reading them from members of the community, and then attempting to answer them. So please feel free at this time to enter questions into the Q&A session of this. And before I close and go into questions, we're very, very interested in having collaborators in this project. So I encourage any of you who all might be interested in this to drop me a note at Kenneth Buetow at asu.edu, and we'd welcome the chance to collaborate and continue this conversation. So what I'll do is open the questions here and see if we actually have any incoming questions at this point. Again, I'd ask you all to please enter your questions in the question and answer box. Looking forward to the opportunity to more directly engage with you all. So if you have any specific questions, I welcome the chance to address those questions. I can see that someone has indicated they're having a little bit of trouble using the question box. It's saying that they can enter questions, but they can't save them. I think that look through, I'd encourage you to try yet again if perhaps if you have specific questions. I would welcome the chance to see if there's that I could answer anything that folks have. I think that this is an interesting and exciting area that permits us to conceivably look at and address particular questions. We're happy to share out. I believe one of the questions that's raised is can the PowerPoints be shared? I believe they're available. The whole webcast will be available, and we're happy if you drop me a note. I'm actually happy to send them to you all. Would be happy to do what I might do to be able to share the material with you. So actually I'm happy to share, and I can see the question related about saving had to do with, or entering had to do with actually slides. So are there any questions that I can ask or that the community has that I could answer related to the particular talk? So I can see that I have a question here relating to, let me start with the first question. So it says, let me look at the full question. If I look at this question, it says that the question says, let me go back, sorry, incoming questions. If I look at the questions that are before me, let's see. So it says, you think researchers could be planning projects where they could collect experimental data and compare it using these tools? And the short answer to this question is absolutely. And I think part of what we're attempting to do is create an open source infrastructure that would permit one to approach these problems on a large scale, and we would look forward to working with anybody, both in sharing out the specific tooling that we've created, as well as bringing in specific data. Okay, I have a question related to the, is it possible to understand calcium signaling in cancer? So as I pointed out in the breast cancer analysis, in fact, the calcium signature allows us to integrate what's been described on an individual locus by locus basis, then what we can do is dissect based on finding that this particular signaling pathway is important, find which are the individual components and or which are the individual variants and how they combine to produce this particular outcome. Are there other questions? So I'll give folks a minute or two more. We have another minute or so before we're formally over. What I would like to suggest, just in closing, is that if there are no additional questions, that I point you all to the obtaining CME credits or continuing medical education or get continuing education credits or continuing medical education credits. Go to the address shown on the slide here, www.bioconferencelive.com, and you can actually take the test and go through and get official credit for participating in this lecture and hearing the results. Again, I'd like to thank you all for your time. I'm welcome individual contacts if folks want to touch me directly. It's possible to download these slides by going in the contents button of your screen and to be able to capture the information. And as well as I know the webcast will be made available shortly. So thank you all for your time. Please feel free to go to the Continuing Education's credit locale. And with that, I say thank you.